Hello, and welcome to the Boffin Buzz, the podcast for anyone fascinated by farming's future. So welcome to the first Boffin Buzz podcast, and I'm absolutely delighted to be joined today by Dr. Tom Thirkell of the Crop Science Centre. Now, one of the things that I think is really fascinating as a farmer, as I walk through my crops, is, I can see the crops themselves, what they're doing, how they're growing, and so on. But what I don't know is what's going on beneath the soil surface. And this is an area that Tom is a bit of an expert in terms of arbuscular mycorrhizal fungi. And everyone talks about this arbuscular mycorrhizal fungi, don't they, Tom? But not a lot of us know an awful lot about it. So can you tell me what it is?
Yeah, of course. So it's this soil-dwelling fungi that is completely below ground. It doesn't produce any mushrooms or any above ground structures. But what it does do is grow really extensively through the soil. And it interacts with the plant roots. So it exchanges nutrients. It gets phosphorus and nitrogen and water and other nutrients from the soil, gives those to the plant. And then in exchange, it gets carbon from the plant. And this is obviously really important in crop systems where we're looking for nutrient uptake.
I see. So where's this come from, this relationship? Has it always been there?
Yeah, definitely has. It's incredibly ancient. It's currently thought that plants sort of colonize the land by using these fungi. It goes all that far back to 450 million years. So by forming this relationship with the fungi, the plants could get nutrients from early soils and not just aquatic systems. So yeah, it's always been there.
Right. So you mentioned it just a minute ago that there's this this relationship. So as I understand that the fungi take up nutrients from the soil, pass it to the plant, and then the plant passes back. It's carbon to the to the fungi, does it?
It's some sugars and also some lipids, and it's been found out quite recently that the fungus can't actually make those lipids itself, but it does need them to survive. And so that fact means that these fungi can't survive without a plant host. So they need the plant to get that crucial carbon supply of lipids.
And is that all plants?
It's a majority of plants, certainly. About 90% of plant species, I believe, form mycorrhizas of some sort, and about 70% form arbuscular mycorrhizal symbiosis. And included in that 70% are all of the top 10 crops. So, very, very clear importance in agriculture.
Right. Are there any crops that don't make that relationship then that you know of?
Yeah, the brassicas and the beet crops don't form as mycorrhizas, and it's not totally clear why. They probably have different phosphorus uptake mechanisms, whether that's exuding acids or interactions with other soil microbes, bacteria, to get their phosphorus. But most crops, certainly all the cereals, legumes.
Right, I see. So now everyone is always talking about soil health. It's almost sort of drummed into us on a daily basis that we have to think about soil health and so on and so forth. So when it comes to AMF, then Tom, what is the relation? What's the significance here with soil health?
Their mycorrhizal fungi, that is, are really important. They've been called ecosystem engineers or keystone species, and that's because they have a sort of a massive impact, even more than their abundance would suggest, on how the soil supports plants. And having said they do more than their biomass would suggest, that's a huge amount, because these mycorrhizas can make up about 50% of the microbial biomass in soils. Right. Can be these arbuscular mycorrhizal fungi, so huge players. And I think if we are getting into researching soils and getting the most out of soil microbes, arbuscular mycorrhiza is definitely the place to start.
I see, and just what is the significance in this relationship? I mean, as I understand it, the fungi is completely dependent on the plant. I mean, it sounds like a sort of controlling coercive behaviour here, almost. I mean, is it entirely up to the plant as to when that relationship is switched on and off?
Well, the short and disappointing answer is we just don't know. The fungus definitely needs the plant for its whole life cycle. The extent to which the plant needs the fungus is much less clear. All the time these crops are being bathed in nutrients, the amount that the plants need this fungus is probably actually fairly low. But looking forward to where we're looking to reduce nutrient inputs from bagged fertilisers, this is where we're hoping these soil microbes can sort of fill that gap and take up the slack and provide those nutrients that are, very often there in the soil, phosphorus especially, but just getting it up into the plants.
Well, that's really interesting because, of course, the other thing that we're constantly being told is that we're polluting the soils with our nitrogen and our phosphorus and everything else that we plaster it with, and so I'm just thinking to myself, all these years I've been putting all of these bagged fertilizers and bagged inputs on the soil. What have I been doing to the mycorrhizal fungi? Have I been killing it? Have I been suppressing it? Perhaps I should be doing more to encourage it.
Well, yeah, it's you won't have killed it. It's definitely still there.
Oh, that's relief.
Don't want to give away any spoilers, but data we got from this project suggesting that you've definitely not killed it off. But I think almost certainly, the broad trends are the more disturbance means that you do to your soil means the more sort of suppressive that's going to be on your mycorrhizas, whether that's
right.
Chemical disturbance or physical. So, yeah, I think soils that have had a lot of bagged nutrients going onto them are going to have probably lower abundance and diversity of these mycorrhizas.
And what's the difference there? I mean, is it the bagged fertilizer that makes the difference, or is it the cultivations? What has the biggest influence on the population?
I'm not really sure, to be honest, and I don't know that anyone's certain about that. It's really difficult to test this sort of thing because you need to say, right, this block of land we're going to treat in this way for the next 20 years, and this bit we're going to do this way. So what you find is a lot of sort of association studies and surveys where you sample from a huge range of places, you take their information, and then you try and tease out the differences. And what that has shown is that, as you might expect, fungicides are pretty bad for fungal diversity in the soil, and at the same time nutrient inputs. And so again, it's just the more you do, the more you put on, whether that is a pesticide or a fertilizer, that's not great for your soil microbes.
Right. Okay. Well, now we've been doing a project on this, and we'll talk about that in a minute. And also, you've got some results from my farm as well, which I haven't yet seen, so I'm really keen to know about that. But we're going to keep everyone in suspense for the moment on that, because I want to ask you about why you're doing this work, Tom. And I should really have introduced you at the start. So could you just sort of tell a little bit about who the Crop Science Centre are and the work that you're doing, because I know that you're involved in this really fascinating, groundbreaking project.
Mm-hmm. Yeah, of course. So Crop Science Centre is a fairly new institute, which is collaborative. It's a sort of union of the University of Cambridge and NIAB here in Cambridge, and we're looking at taking fundamental sort of blue skies research in the lab out into the field, and so a more sort of translational approach so that we can bridge that gap between lab scientists and field scientists, and we can sort of achieve a lot more together is the ambition. And the work that I'm focusing on at the moment, we're looking at soil microbes, and as I briefly mentioned, the way we can use those to plug that gap in nutrient delivery into plants. We're relying less on fertiliser inputs and more on what's already there in the soil, what we can put in with microbes and then what those microbes can get out and put into the plants. And
I think you're working, if I remember rightly, on potentially nitrogen-fixing wheat. Is that right?
that's it, yeah. That's one of the big goals that we're working towards is nitrogen-fixing cereals, yeah. So it's working out how legumes accommodate bacteria in their roots, form these special nodule structures, and then how the plants regulate that so that they deliver enough carbon to keep the bacteria happy, and the bacteria deliver enough nitrogen to keep the plants happy. And it's a huge challenge, but there's certainly progress being made. It's all very exciting.
As I understand it, though, I mean, so we were talking about mycorrhizal fungi a minute ago. That's one relationship, but what little I know about the relationship that legumes have with nitrogen is that they've got this. It's to do with the relationship with bacteria. That's two completely different species. On the one hand, we're talking fungi, and on the other hand, we're talking bacteria. Why are you looking at fungi within this project, where essentially it's bacteria that you should be looking at?
Yeah, so this is an extension of that original project, and it's sort of aligned with the nodulating cereals because it was found out that the genes that control some of the aspects of nodulation also control the mycorrhizal symbiosis.
Right.
And that then launched a whole new branch of research. And so now in the lab and in the glasshouse, we've got really good control over mycorrhizal colonization of cereal crops. So this is gene editing and genetic modification. We can either turn off that association, so there's no mycorrhizas, or we can make it hyper-colonized, so the roots are just completely jam-packed full of fungi.
Right, well, you have just switched on an interest nodule in my head, because gene editing is something that I've certainly got a passion for, Tom. And so what you're saying is that we can actually engineer this aspect within our plants. We can switch it off with gene editing. We can make it enhanced with GM. So are we saying that we could have plants in the future that have that are genetically engineered or whatever to enhance this relationship?
That's exactly it. That's what we're looking to do. We're looking to optimize the amount of colonization. It's in annual crops especially, what you've got is this cycle where at germination there's no colonization, obviously, because there's very little root material in the ground, and then the roots have got to become colonized quickly so that the nutrient exchange can occur and the plants can get the benefit of these fungi. So it's working out how to optimize the levels of colonization. And where this ties in with our farmer participation with the boffin work is that we can create, let's say hypothetically, we can create this perfect plant that cooperates perfectly with the mycorrhizas, but those plants have then got to go into the right soils that have got a suitable mycorrhizal community there that's ready to receive those plants, if you like, and engage with them, and then get the benefit from that symbiosis. If they go into the wrong soils, then they're not going to get any benefit because there's not a helpful community there for them.
Yeah. So just you touched on the project that we've been doing. So do you want to just introduce that project? And what we're trying to find out from this?
Yeah, of course. Yeah, we were looking at the mycorrhizal colonization in different wheat varieties, and what I wanted to know really was across the country and across different management styles, how does mycorrhizal colonization differ? What patterns do we see? Are there trends that different management cause different colonization patterns? And so with yourselves at Boffin, we sort of recruited as many farmers as we could, and we have, I think, 42 farmers sending in samples, and they came back to the lab and, sorry, when I say samples, I mean root samples.
Yeah, well, just to give everyone some background here, I mean, so Tom got in touch about this project and said, "Do you think Boffin farmers would be interested?" I was pretty sure that they would be, and oh boy, they were. So just to give you some background, we've now got about 550 members, of which about 300 are farmers, and we had a lot of interest in this right across the board of our membership, and so we had I think it was about 140 of the members signed up to be involved in this, of which 79, I think it was, were farmers who were really interested in doing this. And as you say, 42 farmers actually sent in some root samples. So if you remember, Tom, we came up with a protocol for the actual sampling of this. Do you want to just sort of talk through that protocol, what the farmers had to do?
Yeah, of course. Yeah. So to get this sort of variation across the country and through different farmers, that relies on having a standard protocol so that everyone follows the same instructions and all the samples arrive to us in sort of similar, coming from similar starting point. So what we asked our farmers to do is to dig up at least 3 wheat plants from their field, and then to wash the soil off of the roots, clean the roots, dry the roots, and then cut them up, these roots from these 3 plants, mix them, and then separate them. So some went into a wet sample tube, and that has been taken, and we've counted the mycorrhizal colonisation in those. So we've quantified the amount of mycorrhizal fungi in those roots. And then the other stayed dry. They are currently waiting in the freezer, and from those we're going to extract DNA, and we're going to do DNA sequencing so we can get an idea of the microbial community. So in addition to the amount of mycorrhizal fungi, we're going to get the sort of the composition, the makeup of that community, so which species are there. And I think that's going to be really interesting. You'll see later when we talk through some data.
Yeah, well, before we get to the data, Tom, I just wanted to say a bit about the methodology. I mean, so I did it on my farm, and I have to say it was quite a hassle. It's not the easiest thing to do, to dig up roots correctly and then wash them and so on. And judging from the Twitter traffic and comments and so on that we got, I wasn't the only farmer who found it quite an interesting challenge, if you like, to do. But tell me about the samples that you got in, because I believe all of us Boffin members stepped up to the plate, I believe.
Yeah, absolutely. I was pleasantly surprised, I'll say. All of the samples arrived. It's just perfect condition, exactly as the protocol requested, and all in the right tubes, the tubes in the envelopes, all great. The only thing I did to all of the tubes, just to be sure, was to replace the storage solution, because some of that may have evaporated in the post, so I just wanted to check that, just make sure that was all still good. Otherwise, absolutely perfect. It's really exciting.
Yeah, no, well, I think that's a really important part of this, actually, Tom, because one criticism that we always get at Boffin is that, well, farmers can't do proper scientific trials. It's not like small plot work and so on. Well, I think we've shown here that that actually farmers can do proper scientific trials. They can follow a scientific protocol. And by the way, I might just pick you up on one word that you used there, that you were surprised. I don't think you should be surprised. We're talking about some of the best farmers in the country as Boffin members doing this. But it's really good to hear, actually, that you got some really good data. And I think everyone filled in their sheets as well, didn't they?
Yes, yeah. So we collected information. We asked participants to send in their details for cropping history and management styles and inputs. So we've got all of that information as well that we can use to compare, and it was all really informative as well.
Yeah, that's quite an important part of the project, I understand, and to understand what the nature of the farming system is and relating that to the actual mycorrhizal fungi. Was that the idea?
Absolutely, yeah,
Right.
absolutely. Because without this information, we've just got 42 sets of numbers, which we can't pull any useful insights from. But with the information that's been put on this questionnaire, we've also got that, so we can see the different trends between managements and so on. Yeah.
Great. Okay. Well, look, going into the actual results themselves, Tom, I believe that we're going to be shortly sending out the results to the farmers who took part. So, what will they actually receive when they get their data through?
They can receive an information pack, so they'll get not only the amounts of fungi in their roots, but they'll also see how that compares to all of the other samples we collected. And so when I say the amounts of fungi, we've got 3 different measures that we've taken. We've got the total mycorrhizal colonization, so the percentage of your root system that's got mycorrhizal fungi in it, of any sort of kind. And then we break it down a bit more detailed, so we've got special structures inside the roots that the fungus forms. And these are arbuscules, and they are really highly branched, and hopefully we can put up a picture of these to show on the video. Really highly branched structures, and the branching increases the surface area, and that surface area means that you can get a lot more nutrient exchange between the plant and the fungus. So at these arbuscules, that's where the plant is providing phosphorus and nitrogen. Sorry, the fungus is providing phosphorus and nitrogen to the plant, and the plant is providing carbon as sugars and lipids to the fungus. So,
just to explain to those people who are watching this, we've got a picture up at the moment of an arbuscle, which is what Tom's talking about, and that's where the arbuscular mycorrhizal fungi comes from, presumably. I imagine and that is so. Is that the point at which the plant communicates with the fungus? That is the epicenter of this relationship, I guess, isn't it?
That's where, certainly a lot of communication going on there.
Yeah.
The plant and the fungus are actually communicating everywhere all the time, pretty much. They're in communication before they even make physical contact in the soil.
I see.
But
So the data that farmers get through, the first bit is about the percentage within the roots of Arbuscular mycorrhizal fungi, and then the second one is so is that percentage of arbuscles?
Yeah, so the data is all presented the same sort of way. It's the percentage of the root system that's got these structures in it, and we do that by looking down the microscope and looking at a very, very small part of the root and quantifying whether or not these structures are present. And so in itself, it may not be hugely informative as an isolated number. So this is where it's important that we compare it to different treatments, direct drilling or no-till or these sorts of things.
Right, and the farmers will get an idea from this data of where they sit in the spectrum, if you like.
Yes, exactly.
So let's not delve too much into that, and reveal too many secrets, because we've got the data in front of, or at least Tom and I have got the data. We're not going to obviously share it with everyone, but there is another column on here. Is that vesicles or something?
Vesicles. Yeah, they are more fungal structures in the root, and they are storage organs for the fungus. And they're normally just jam-packed full of these lipids that the fungus is receiving from the plant. And so you get lots of sort of hints and ideas of what the fungus is doing and what the plant's doing by the numbers of total colonization, arbuscules and vesicles. And what you'll very often find is that when the plant's growing, its fastest growth stages, that's when you get lots of arbuscules, because that's when the plants and the fungi are exchanging lots of nutrients. What happens later in the year is that the fungus is getting signals that the carbon supply is slowing down, and with an annual plant like a cereal, the carbon supply is very quickly going to stop, And so the fungus sort of switches into carbon saving and storage. So rather than foraging nutrients, the fungus is then packing these vesicles full of lipids. So it's got that carbon supply, because once the plant's dead, the fungus can't get any more carbon. And in an annual system especially, this is really important because it allows that fungus to survive and produce spores through that time when there's no living roots in the soil.
Yeah, just thinking from climate change point of view and so on, that's also a really important time for carbon capture and storage, I imagine. I mean, because we're talking about this is the mechanism through which our soils store carbon. It sounds like. So is that the case? Is that the point at which you talked about this carbon storage that the fungi is doing? I mean, that's the holy grail for us as farmers, isn't it? That's what we're looking to do.
Yes and no. I mean, it's short-term carbon storage, and so every year, you're absolutely right. Every year, when the plants are growing fast, there's also a huge amount of carbon going down underground. And there's a recent paper, I can't remember the exact group who did it, sorry, but I think they calculated that something like 1/3 of annual carbon emissions from, anthropological sources can be, the equivalent 1/3 of human emissions is sent into mycorrhizal fungi every year. So
1/3?
Enormous amounts of carbon. As I say, because of its nature, I think best understanding at the moment is that that's short-term storage, and from a climate change perspective, what we want is long-term storage. We want it to go there and stay there.
Right.
But that doesn't take away from the fact that for it to stay there, it's got to go there first. And so, mechanisms that are putting it into the soil, like mycorrhizas, got to look into that as a mechanism for sure.
Right, I see. So going back to the arbuscles and the vesicles and so on and so forth, and looking at your data that you get through, I mean, presumably high percentages of all is good. Is there a particular one of those where you should be, really trying to focus your attention on getting the number up or whatever? And is there much you can do to actually influence those numbers?
Yeah. It's a good question. I don't know how much you want to go into the data now, but it's just banging the same drum, I think. So what I've got is that it's arbuscules, I think, that we want to be looking at, because they're a really good sign that the fungus is actually being beneficial to the plant, and it forms these so that it can trade nutrients. In other words, so that it can provide phosphorus to the plant and nitrogen. And so it's the arbuscule numbers, I think, that are most important. And what we're seeing here really nicely sort of backs up the received wisdom and what principles of regenerative agriculture, really, that you're lowering disturbance and you've got roots in the soil for as long as possible. So
right.
We need more arbuscules where there's less disturbance, especially in the tillage treatments.
Right, I see. So if you looking at the results, sort of on a broad basis, Tom, you've done a bit of analysis, have you, as to the sort of management systems that are getting the higher percentages? Have you?
Yeah, exactly. And it's really clearly direct drill is really good for mycorrhizal colonisation numbers. And that's
right.
Perhaps not surprising. You're turning the soil over mechanically. You're going to be ripping up all these fungal filaments, the mycelia, in the soil. You then plant into that. Those fungi are going to have a more difficult time re-establishing a network and growing into the roots than a fungal system that's just not been mechanically disturbed in the same way. So direct drill there makes it easier for your plants to get colonised.
Okay, I see. So would you be so bold as to say that it's the tillage methodology that is like the overriding factor here?
From this, no, I wouldn't be as bold as to say that, but only because we haven't collected huge amounts more data on inputs and everything. I think this is an absolutely fantastic starting point, and it's giving us all the clues that this is what we need to be looking at. I think we just need to go further and do more. Inevitably, I was going to say that. But.
So what I'm keen to know is, because we had some organic farmers included in the survey as well. So how did the organic versus conventional? Well, I think we asked people whether they were organic, regenerative, or conventional. What was how did that play out?
That was really interesting. We only had 3 organic farmers, so it's quite a small number, in the whole block. But what that showed was that organic had much higher colonisation levels,
really,
than people who identified as doing conventional management on their farm, and the regenerative as well. So it's really interesting. I think something important to point out, though, is that we're trying to get the most out of our mycorrhizas, but they're not some magic silver bullet that's going to solve all these problems. It's something that is definitely important to pay attention to, but I'm not going to say now, if we optimize our mycorrhizas, you can get high input yields from no inputs, and the mycorrhizas are just going to give you all of your nitrogen and phosphorus you ever need to get, record-breaking yields. I don't think that's realistic, but they can certainly do a lot.
Yeah.
And organic seems to be a great way to get the most colonisation.
Well, I guess it makes sense, really, because the organic guys are looking to draw in from resources that they're not applying to their fields, and so and they're going to be an AMF is an obvious route to drawing in that resource. So it's natural that relationship is working. And I suppose the regen guys, that's the route that they're heading towards as well. So there's obviously a natural progression there. That's interesting. Now, one thing I wanted to point out is one thing I should have said is that we're very grateful to KWS UK and RAGT Seeds, who have very kindly helped support this project. And I think you did some extra work with with KWS and RAGT.
Yeah.
Do you want to just talk us through that?
Yeah. So conveniently for me, they both have sites very close to Cambridge, so I could get to there and sample from their trials. They have obviously a whole range of stuff that they're growing. At RAGT, they've got lots of different plots where they're testing different cover cropping mechanisms and approaches, and they didn't actually tell me what they were, so I've got numbers for different cover cropping regimes, but I don't know what they are. So I've got 1 to 6, I think it is, of their different cover crops, and compared to a fallow, and that's fascinating. I think there's 3 times as much mycorrhizal fungi in Skyfall if that ground had a cover crop on it before, compared to in Skyfall that went into what was previously fallow. So
really, hang on. Let me just repeat that. So if you're putting Skyfall into land that has just had a cover crop, you get 3 times as much AMF than if it just went into bare soil.
Yeah, exactly.
Wow, that's
I think
fascinating.
It's everything that everyone's been suggesting. That the longer you've got those living roots in the soil, the longer you've got carbon being provided to the mycorrhiza, and it can stay in that growing, foraging sort of growth habit. If you've got a fallow period, you've got no living roots, you've got fungi that are retreating into spores and being dormant, and then they're just waiting for another root to come along, which would be your Skyfall that goes in in the autumn. And so I just think it was amazing. Really,
and is this relationship, has it got anything to do with Skyfall as such? I mean, is it, do you get it with X days and other varieties?
I don't know. I haven't checked that. But it's not part of this work. But it's you see it in the data. Yeah, so it's all this stuff you have to piece together from little studies or little bits of studies that have been done all around the world and everywhere. So yeah, it's is that cover cropping is just generally good for mycorrhizas.
I see. Great. So that
The
those
RAGT.
sorry.
Sorry, I was going to say the RAGT was just skyfall, so that was all I could sample from there.
Was there anything in particular that you found from the KWS results?
Yeah, the KWS had a site. They were growing a range of different wheat varieties, and they were testing spring sown versus winter sown. I think at some point to demonstrate the importance of fertilisation, but also just to compare those the growth there. Really interestingly, at the time we sampled, which I believe was the 1st of June this year, there's no difference between spring sown and winter sown crops in terms of their colonisation. But what we did see was huge variation between the different cultivars or the varieties that they grew.
Really?
They had 7 different varieties, and they ranged hugely, something like 20 to almost 50% of the colonisation in the roots, sorry. So colonisation varied from about 20%, about 50%, just depending on the cultivar that you've got. Otherwise, everything exactly the same.
Okay, so I don't know whether we're going to be able to broadcast this, Tom. We'll have to get permission. But can you tell me some individual varieties that performed well and those that didn't?
Ooh, yes. So, if I just get the data, sorry, I don't have it to hand. X-Days was the highest at 48.8%, and then Palladium and Xylem and Ladum were much lower, so they're around 25-30%.
Oh, interesting. Right.
And I know you see the same with the arbuscules as well. So X-Days has got the highest number of arbuscules by a long way, 13%, and then we've got Palladium 5% arbuscules.
Right.
So something's going on where Palladium doesn't think it needs mycorrhizas in the same way that X-Days does. And I should say, because these mycorrhizas are absolutely everywhere in the soils, we don't know the impact these mycorrhizas are having on these different varieties, or indeed on our farmers' samples. So what I really want to do is test these in the glass house, where we can have sort of soil that you sterilise, and then you either put the mycorrhizas or other microbes back in, or you don't, and then you can compare how do those crops do with the mycorrhizas versus without.
Right.
Because it might be that those ones that have got really high colonisation are sort of allowing a lot more colonisation because they need that interaction, or they get more benefit from it. And the ones with really low colonisation, they're not getting a benefit from it. It may be root architecture can have a big impact on that, and a whole load of other factors. So, as well as our management styles and our plant genetics in terms of those signalling, we've also got this underlying, all these other factors, root architecture and so on, that can also impact our mycorrhizas.
Oh, great. Well, we'll come to that in a moment, Tom, because I'm dying to know my own results. And as I say, I haven't seen them yet, so I don't know how I fare. So come on, tell me.
You want the news? So we've got, it was Thirkell, I believe, I sampled.
That's right, yeah. So I guess that's one thing to point out before we... So we had asked everyone to sample their wheat crop, and specifically we asked if they had it, to do either X-Days or Skyfall and a first wheat crop, so that we got consistent results all the way through. So I guess the first thing to say is that I wasn't actually growing any wheat this year. I was just growing Thirkell. So mine was actually done in a Thirkell crop. So I guess that's one thing to point out first of all.
Yeah. And it was also a fair bit earlier, wasn't it? Because we wanted to have a demonstration of what our participants' samples would look like and how to.
Yeah, I get the feeling, Tom, that you're building me up to something that tells me I've got crashingly low results.
No, not crashingly low at all. Lower than average, certainly. So,
okay.
there was 18% colonisation in total.
18?
2%, yes, 18. And then 2% arbuscules.
Okay.
And we found no vesicles at all.
Ah, right.
So just compare those to the averages. So
Yeah.
total colonisation, you had 18. Our average across all of our farmer samples was 34%.
Right.
So about half.
Okay.
And then in arbuscules, the average was 9%.
And I got
had
2.
2%.
Yeah.
And then in vesicles, the average was 2%.
And I got
And
0.
we found 0 in yours.
Yeah.
But
Okay.
again, this is exactly why we wanted, as far as possible, to have the same variety, because what the KWS samples have shown is that variety has a huge impact. So it may not be anything you're doing to your soils. It may just be that Thirkell doesn't have much mycorrhizal fungus in it. So if we'd taken from 15, 20 other farms that are growing Thirkell, you might find that yours is actually surprisingly high.
Right. Okay. So I shouldn't read too much into it.
Don't read too much into individual results, no. But I think what it really demonstrates,
ashes for me or whatever.
It just really demonstrates again the importance of the sort of stuff we've done and how that's the approach that you have to use to get these trends and see what's happening.
Great, I see. Okay, well, and now you just touched on it a moment ago in terms of the significance of this and where we can take it. And so, can I ask first of all, from the point of view of your project with Crop Science Centre that you were talking about earlier, what's the significance of these results for that?
I think it's really important, and it's something I really want to push further, so we make more of a priority of what we're doing here, is bringing in more of a soils focus, so that we're not just looking at the minute plant genetics aspect, we're also looking at the sorts of soils these plants are going into, and that means collaborating with a huge number of farmers, because that's where the soils are. And so I think there's enormous potential here for expanding this work and also, picking the low-hanging fruit so that we can see big differences and then drilling further into those. And that would mean work not only on farm but also back in the glass houses here as well.
And then the significance for the farmers themselves. I mean, I'm just wondering what I can take away from this. And initially I was thinking, oh my God, I've got to up my game as far as my regenerative practices are concerned. But we don't practice cover cropping. It's worth noting. And perhaps if we did, we'd get more colonisation. But then I shouldn't really be reading too much into these results because, as you said, it's Thirkell versus wheat, which could in itself be misleading. So,
Yep.
What's the significance of this for the farmers? Well, especially our farmers who are actually involved in the study. What can they take away from this?
I think a few things. I mean, most excitingly, I think it shows that the system works. That farmers can provide really robust, good data that is really helpful and useful, and it's working together on these problems, which we all need to do. And I think as well, exciting, it demonstrates that the management is having an impact, and the choices made on farm are having big influences on what's going on below ground as well as above ground. And that makes everything more complicated but also more exciting. So
yeah. Well, I'm glad you say that, Tom, because as you know, we've been working at Boffin towards what we're calling the Truth Project. So this is thriving roots underpin total soil health. There's a stray S in there, so it doesn't quite work out. But there we go. We're looking about the truth to do with our soils, and this is a project that we're currently looking for funding for, and it's to do with this whole relationship that plant roots have with the soil, and it's not just down to mycorrhizal fungi. It's down to a whole other range of other factors as well, and we're looking specifically also at the genetics of this. So there's some really interesting work that's been going on at John Innes Centre, for example, the Wish Roots project, where they've been looking at some of these old landrace varieties, and also durum wheat and other sort of wheats like that, wheats that aren't commonly grown, and looking at the root architecture. What we're hoping to do is get a project underway, if we can find the funding for it, where we can get farmers involved in this work, get a proper protocol to actually study the rooting that's there, and also study the soils and the relationship. When I say the soils, the microbiome content and things like that, and then look at that relationship between the rooting architecture and the rooting structure and so on, and the microbiome. So that's something that we're working towards, and hopefully, we'll get a project underway. But I mean, the importance of what we're looking to do here, Tom, is get farmers involved in that work, and the end game here is a platform that we can set up where we can. We've got a number of farmers who have got this protocol so that they can measure it. Measure what they're doing. Actually, get some proper measurements for not only the rooting but also the microbiome content as well. And then we can look at how we can influence that. What we can do to improve the rooting, what we can do to improve soil health, and so on. Perhaps using inputs or genetics or whatever to do that. So I think this is a project that you'd like to be involved in, isn't it, Tom?
Absolutely. Yeah. I'm already committed to doing some mycorrhizal analysis for you all, if provided we get funding. Fingers crossed.
Yep, absolutely. Well, we have put an application in, and so we're just waiting to hear back on it. But it is a project that we want to take forward, and I know that a lot of our members are really, really interested in this area. But look, we've been rabbiting on for ages about this, and I think it's probably time to draw this to a close. But this is a topic that we will definitely return to shortly. Just want to reiterate, this is the first of our podcast, of the Boffin Buzz podcast. Do subscribe to it, and as I say, we'll be coming back to this topic. We'll be covering all sorts of other topics as well that are looking at our other projects that we're doing. We've got some exciting work going on in pulses. We've also got some groundbreaking work going on in slugs. Harvest weed seed control is another big focus area for us as well. So we'll be exploring all of these areas in the coming weeks. But Tom, will you come back at some point? Perhaps later in the year or next year, and when we've got some more sort of information to impart with everyone, and come and give us an update.
Yeah, of course, absolutely, love to. I'll hopefully have all the DNA sequencing results back then as well, so I can bombard people with data and trends and everything, and we can see what's going on.
Yes, of course, absolutely. And just to let those farmers know who have submitted their samples, so this information will be coming out to you shortly. We'll draw the information together, and that'll come out to you shortly. And then the DNA sampling results, whenabouts do we think that's going to be available, Tom?
Oh, that's a very good question. I don't know, is the short answer to that. It relies on a whole bunch of boring technical things, and also fit them onto a batch of analysis. So we'll just see our GP pull up here and see if we can get people sending samples off soon, so that we can get these sent off and processed. But early next year, definitely, hopefully.
Right. Okay. Well, we'll keep everyone informed as far as that's concerned. Yep. And then once we've got the results back, we'll do the same thing again in terms of making sure that we can not only give people their results, but tell them what they mean as well. So, but look, let's draw things to a close. Tom, thank you so much for giving up your time and talking. It's been a fascinating conversation. I know that this is an area that a lot of our members are really, really interested in. So that's all we've got time for. So thank you very much, Tom. Thank you very much, everyone, for joining us, and see you again soon. Thanks.